Welcome back to the rodent neurosurgery modules. So today's session is pretty important for the safety of the personnel and also for the lab animals handling. So today we'll discuss about standard safety practices. It's very important to prevent acquiring any infection from the animals that you're handling, and try and protecting yourself is of utmost importance when you're handling lab animals. So by and large they are clean, and because obviously they are from labs which are bred for the sake of experiments, but however there are still many diseases which can go unnoticed within the animal, and that can get transmitted to the personnel who is handling the rat, that is you if you are handling these lab animals. So today we will look at what are all the precautions that needs to be followed, and how a sterile field is maintained, and how sterile clothing is worn, and how is it really maintained throughout the procedure, throughout your stay within the lab. So let's look at those aspects in today's session. So first and foremost, you need to be familiar with the term called zoonotic diseases. Those zoonotic diseases are those conditions which are acquired as a result of handling the animals. So zoonotic disease in a broader sense is basically a disease acquired by handling any animals, transmitted from the animal to the humans is zoonotic diseases. And specifically the diseases associated with rodents are these that are listed: rat bite fever, tularemia, hanta virus. There are n number of diseases that you will be prone when you are handling the rodents. So it's very important that you take utmost precautions while handling these animals. However clean they look, however hygienic they look, they can still harbor these organisms, and when they get transmitted to the health personnel, they will definitely cause these kind of diseases, which can manifest with various symptoms. They can have, you will have headache, vomiting, and they will have hemorrhagic manifestation. Then you have meningitis, n number of illnesses with significant symptoms and morbidity. So these are the diseases that you should protect yourself acquiring from the rodents while handling. So how are these transmitted between animals? So it can happen such that the contagious disease between different species of the rats or different species of mice, when they are bred in the animal lab, it can happen that the transmitting agent can become contagious amongst the rat itself. Then individual animals themselves can carry these agents and can directly get transmitted to humans. And then of course animals from different colonies and different locations. So all these are possible sources for the microorganisms to get into the rodent body, and by handling it can get transmitted to the human personnel while handling, if you are not careful with these safety practices. Alright, so there are some general guidelines which need to be followed. So basically you are sort of creating a barrier between yourself and the rodent that you are handling. Alright, so the direct contact is what we are trying to avoid by following these guidelines. So there are two things. One is just the protective gear that you need to wear while handling the rodents. The next is that you need to maintain, if suppose you are going for surgery, that is another aspect of today's session, where when you are going to do the rodent neurosurgery or rodent any surgery for that matter, it is also important that you do not contaminate the field and transmit some microorganisms into the body of the rodent, which will affect your experimental outcomes. So the crossing of these microorganisms can happen both ways, and it is really important that you try and curtail both the ways of transmission from rodent to humans and from humans to rodent, or for that matter from the atmosphere or the environment that you are handling the rodent, that can also cause transmission of agents into the rodent's body, and then your experiment had to stop in midway because of the dreaded infection that the rodent has acquired during the surgery. So that's about how do you maintain sterility while handling the animal so that no infectious agent can get into the animal's body. Okay, that's another thing. So the first and foremost is obviously, as you can see here, is the lab coat that needs to be worn over the street clothes when you are entering the animal housing room or whenever you are handling the animal. So there are possibility that the animal hair itself is enough that can stick to your regular street clothes and then can transmit, you carry over into your dining areas or even your living areas, then those can harbor organisms and then transmit the disease which the rodent was having. So naturally the lab coat is at most important. If you're not really doing a surgery, if you're just handling, so then lab coat is what is enough. So there are two different ways of looking at it. So are you going to wear a sterile apron, or you're going to wear just a lab coat which is going to be a barrier between you and the animal, as I said earlier. So that depends what sort of situation that you're really handling the animal. If you're going to handle the animal in terms of surgery, then you might as well wear a clean gowns and clean gloves until you actually start the surgery, and then just before the surgery you change these unsterile, just clean, but then they are not completely sterile. They can have few organisms, but you're just handling outside the rodent body. You're not making an incision, you're not cutting, you're not really starting any surgery. So that sort of handling, where you're trying to position the rodent onto your OT table or on the stereotactic apparatus, you're just positioning it, you're trying to shave the hair, all that requires these kind of generally clean lab coat and then the clean gloves. That should be followed to prevent your body contact with the body fluid of the rodent or the organism that the rodent skin can harbor. And of course eye protection, if you are trying to sort of use some fluids to clean the rat or some chemical disinfectants that you are going to clean the area which you are going to perform the surgery, then these sort of mask and gowns are also required. So these are the general lab attire that you need to follow before entering the lab. So starting from top, you need to have a cap, a face mask, and if possible the face shield, and then a good respirator mask, and then of course the gown or apron and the gloves. So these are the minimal requirements that you need to use before entering the animal facility. This is very, very important. If you're just going to, as I said, going to generally handle the animal, and maybe you're doing a behavioral experiments where you're going to observe some behavioral changes and you're going to note down those behavioral changes, then this should suffice. If suppose in addition you are going to do the surgery after you enter, then this cloth should be substituted with sterile gown and sterile gloves, and entire sterile accessories needs to be worn. All right, so that is something to observe. Then obviously try and avoid taking any of your personal articles like food items, books, combs, hats. So just leave everything outside the animal room and try and avoid taking any sort of articles inside the lab. If there are any stationaries that are required, if you need any accessory that needs to be used in animal lab, maybe once you take it and leave it there until your experiment is over, until that methodology is completed, or until your study is over. So that would safeguard from taking these articles which can harbor these organisms which we just described from coming outside. So you leave the articles and then let that be discarded at the end of your study. So use some stationaries which can be discarded, or at least those can be sterilized using alcohol, I mean ethanol or formalin at the end of your experiment. So it's very important that you try and avoid taking the personal articles inside the lab. And as I said, there are uniform, there are scrub suits, and there are soft sole shoes or slippers which are dedicated to the facility. So this can be used, and then of course any protective clothing required must be worn by the animal care personnel while in the care room. Okay, so that's very important. So the dresses which are dedicated has to be worn, and then avoid your personal articles from entering the animal facility. So let's come to the situation where you're going to sort of going to do the surgery. Alright, so since this is a surgical module, I thought we will cover the surgical scrubbing as well, which is very important, because if you're since this is a neural engineering course, lot of implants will be used, and as I said earlier, whenever the implants are used, it is very natural that this foreign body can have harbor organisms on its surface, and then get carried out to the brain, and then a biofilm forms on top of your implant. So this biofilm is nothing but the colony of microorganisms which are growing on the surface of your implant, which is going to cause implant failure, because obviously the healing doesn't take place whenever there is infection by various pathogenic organisms. So it's very important that when you're switching from general animal handling or general animal handling for behavioral setups to the surgical handling, you need to go through all these steps that we are going to discuss in next few slides. So once you're in the lab with the lab coat, you need to remove your gowns and gloves which you used for animal handling. So before you sort of change into the scrub suit, which is the OT dresses or the dresses required for surgery, you need to make sure that all the unsterile steps are finished. So there has to be, or you need to have an assistant, and I would prefer to have a person who can help you with the transition from the unsterile to sterile. It is always good to have, and you can help each other in those particular steps where you sort of switch from unsterile to sterile. For example, once you wear your sterile gloves and gowns, then you realize that there is something that needs to be really handled which is in an unsterile area, and then that person can help you with it. So from now onwards, whatever the steps that's going to come, it has to go in one direction. It should never be that you will come back and do something else that needs to be. So the sequence is very important, and the sequence is important to ensure that ultimately when you touch the sterile field or the sterile area, after you open the skin, everything that you see is considered sterile. Alright, you make an incision, from there onwards you are not supposed to touch any unsterile articles. So, and then of course you are making sure when you go in one particular sequence, you are making sure the ultimate gloves that you are going to handle, that should not have harbor any microorganisms on its surface. Alright, that's the end outcome that you are going to keep in mind when you follow these steps. Alright, so first and foremost you wet your hands. So how you really scrub is utmost importance, and how much time you are going to spend time is also important, because unfortunately the microorganisms are really adherent to the biological surfaces of the skin, and skin itself can harbor many organisms which can the colonized organisms which are normal for us, but then when it can get into the animal body can cause diseases. So it's very important that we thoroughly clean with the disinfectant, and there is a way how this hand scrubbing needs to be done. So generally there are these 7 steps of hand washing that is recognized by World Health Organization. It's generally for human purposes and human handling and the patient handling, but then that's a gold standard for maintaining the sterility before you actually wear the OT scrub suit. All right. So once you take the soap onto your hand, you need to rub the palm against each other. That's very important. The center of the palm needs to be rubbed nicely, and then the back of each hand with interlaced fingers. So that is pretty important here, that the web spaces of in between your fingers can harbor a lot of organisms. Alright, so center of the hand first, then you interlace the finger and make sure the web spaces are scrubbed nicely and clean, and making sure entire the web spaces are covered in both the hands. And then of course you rub the palm of the hand first and then the dorsum of the hand. Dorsum is the back side of the hand. Both needs to be covered with the interlaced fingers. Once that is done, then the back of fingers needs to be covered, because the nail can come into picture, and thorough scrubbing of all the portions of the hand is what these 7 steps is to ensure for. And then of course the base of the thumb and the base of fingers, so that also needs to be covered individually. And then comes the tips of your fingers, mainly to clean the fingernail bed. Ideally it should be a good manicure needs to be done, and if that is done, then tip of the finger needs to be rubbed in the center of your palm to ensure that nail bed is taken care of. And finally you rinse and dry your hands. So for a surgical scrub, you need to ensure when you rinse and dry, the water drips along your elbow. You understood? So you need to lift your hand, the hand should be at the topmost level, because when you are going to handle the sterile gloves, it shouldn't be that your tip of your finger will have all the dirty water which has flown from the elbow downwards. So the flow should be away from your hand. So just to give a recap, first is palm, then the interlaced fingers on the palmar surface and the dorsal surface, where you interlace and cover the web spaces. Once that is done, then back of your fingers needs to be covered in the 4th step. Then the 5th step is dedicated for thumb, cleaning the thumb which stands out separately. So the base of the thumb and the web space, which is pretty big, needs to be covered separately. Then of course tip of your fingers for fingernail, and then finally you are going to rinse and dry your hands, with making sure the water flow away from the hand, not towards it. Alright. So that is the standard 7 steps of hand washing. So now let us see, how do you wear this personal protective equipment, for that matter the scrub suit in our scenario, before you go in for surgery. This again, as I said, the sequence is important so that your gloves remains clean and sterile by the time you touch the rodent's sterile field. Alright. So as I said, you will work from the boots where you will use the shoe cover, then you will be washing your hands, then comes you wear the mask and the cap. Then obviously the eye protective gear is also important, because when you do the drilling, it is possible that it can come in contact with your eyes. So all the bone dust can come in, and even for that matter blood particles can also come in contact with your eyes. So it is very important that even the eye cover is taken care of. Once those are worn, then you will do the surgical scrub. As I just mentioned, the hand wash needs to be done thoroughly. After that you will be touching the sterile gown. So in the next few slides I will cover how this gown is worn, and then comes the sterile surgical gloves. So this is, and it is always preferable to use a double surgical gloves. Yes, it is sort of difficult that the double surgical gloves will take away a little bit of your tactile perception. The skin perception does come down, but you will get used to it. But the idea being for wearing the double surgical gloves is that there are chances where the gloves can get torn, or there might be puncture in the gloves to start with. So when these are the instances, and if it's only a single glove, your skin is going to come in contact with all the body fluids. Whereas if there is a double gloves, at least if the outer gloves get damaged during drilling or during any steps of surgery, it's easy to remove that and then wear your second surgical gloves. This is important because there is a sequence to wear the sterile gloves as well, and it becomes easy if you have another gloves inside, alright. Otherwise your assistant has to pull out the gloves and you have to follow the same sequence of wearing the surgical gloves, which we will discuss it in next slide. So this is the entire sequence that you need to follow, so that in the end when you wear the sterile gloves, you are ready to touch the sterile field. That is the whole idea, alright. So it is better you do not skip this particular sequence to ensure that your hands are sterile and clean to proceed with surgery, alright. So now how do you don the sterile gown? Wearing is called donning basically in medical terms. How do you wear the sterile gown? It is very important, because when you use a sterile gown to approach your surgical field, whichever animal, and you are draping the surgical field, and you need to ensure, if you are trying to use microscope, your body is pretty close to the sterile area, alright. And if you are wearing an unsterile gowns, that can contaminate the field. So it is advised that you can wear the sterile gown depending on the demand of the sterility of that procedure. For example, if it is just a small injection that you are going to give into the brain, maybe you can skip the sterile gown. I would strongly recommend sterile gown, but if it is a small procedure for a few minutes, then you can handle it with sterile gloves alone. But if it is a sterile gown, if it is a long procedure and you are going to implant an electronic sensors, or the cortical sensors, and you are going to implant a depth electrode and leave it for a long time, and your study involves a chronic study, then I guess elaborative sterile accessories are required, alright. So anyway, but it is good to know how to you wear the sterile gown, alright. So once you finish scrubbing, all the gowns are folded in such a fashion where you can see the inner and outer surface of the gown. That is the whole idea. So the handling of the gown is always done with the inner surface, not the outer surface. The outer surface is, let me point it out for you. This is what I am referring as outer, this is inner, alright. So the gowns are always folded such that the inner surface is accessible. The outer surface is going to come in contact directly with the sterile field, and that is the reason why you are not going to touch the outer surface with your bare hands before wearing the gloves, because the gloves sequence is going to come only after you wear the sterile gown. So you are coming out from the hand scrubbing area, wipe it clean and your hands are dry, and this is how you hold it. You are holding it such that the inner surface is in contact with your hand. Once you hold that, you drop it so that the gown opens up completely, alright. And while you get more and more surface, inner surface for handling, you need to find out the sleeves, which usually opens up when you drop it that way, holding the inner surface, and then you need to insert the hand into the sleeve following the same inner surface where it comes to the sleeves of the gown, and of course your assistant can pull it back until your hand reaches the area. So that's, and then the ties are provided behind your back, so your gown is ready to use. So the one difference in different practices are that maybe you can avoid bringing your hand out of the sleeves completely and use it to handle the gloves, all right, because gloves are the most sterile part of the entire uh, personal protective equipment or the sterile dresses. So in order to keep the gloves 100% sterile, this is one way of handling it, where you use your hand with the cuffs of your sleeve being held in place, and then you handle it to wear one gloves. Once one of the sterile gloves is worn, the other can be handled easily, all right. So this is bit difficult. I would suggest you can wear the sleeves completely, provided your handling of the gloves is done in a very specific fashion, such as shown here, all right. So the part I missed is the timing for scrubbing. The first scrubbing is always 10 to 15 minutes. So for rodent it is enough that you scrub for 10 minutes and then wipe it clean and you wear the gloves, and this is how you handle it. You will not, again there are surfaces, inner surface and the outer surface. Again the gloves are folded in such a way the inner surface is accessible. Just like what I said for the gown, inner surface of the gloves is accessible to wear it. In order to maintain the sterility, you will be handling only the inner surface, all right. Pick it up and then insert your hand into it, then pull it across your wrist. So there is one difference. I would generally recommend to avoid touching the outer surface yet, all right. Keep it folded, pull it up to the the wrist crease and then leave it there. Now that you have a sterile hand here, insert it into the outer surface of the other gloves, all right. That is the difference. You are not going to touch the inner surface, you are going to insert the space between that fold and then get it onto the other hand. Once the other hand goes in completely, only then you are going to unfold that the pre-folded gloves and cover the entire wrist onto the sleeve of your gown. That is the whole idea, all right. So there will be a sleeve coming till there. This gloves will snap onto the outer surface of the gown, so that the entire your upper limb is covered in a sterile fashion, alright. So this way of donning gloves will ensure that your tip of your fingers and your entire hand remains completely sterile, alright. So then you are ready to start your surgery, provided you have arranged all your equipments in a sterile fashion beforehand, alright. So there is another assistant of yours who will arrange all the equipments if it is possible, or you can arrange it yourself by wearing the gloves, sterile gloves, and keep it ready before you actually go and scrub yourself, alright. Because outside the tray you will not be able to move it, you will not be able to sort of handle things which are unsterile once you wear the surgical gown and surgical gloves. So our entire surgical setup needs to be ready before you go and scrub in, if you really do not have any more help to maintain the sterility. So once the gloves are worn, you are ready to go ahead with the surgery. But apart from it, it is always better to maintain the safety measures which I just said, and you need to wear the gloves always whenever there is a direct contact with animals, especially when you are trying to collect the blood and there are maybe you think that the rat is not really doing great and maybe there is an infection. All the time you need to wear the gloves, alright. And it is very important that the gloves are changed in between the various procedures or steps that you are handling. For example, you are handling the skin, but if you really want to tap blood, change the handling gloves to sterile gloves when you are trying to draw blood. And after the surgery, you again remove the gloves which you use for the surgery, put another pair of gloves which are used for handling, all right. So that sort of measure need to be considered, and you need to make sure that entire scrub suit is discarded while you are leaving the lab, and make sure all those accessories and the suits, scrub suit that you have used, also disposed in a right way. That is very important, all right. So that comes to the end of the session on safety measures, which are very important. To summarize, I would say you have two sets of safety measures here, all right. One is for the animal handling, the next set of measures is for sterility of the surgical field. So both we are tried to cover in this particular session. So that covers the entire surgical aspects and the best safety practices that are needed. In next session we will try and deal with how to perform euthanasia. That is very important, and you need to know the correct methodology of euthanasia, and then how do we really harvest the organs for various lab studies, all right. Thank you all.